Hi, my name is Kristin Branson, and I'm a group leader at the Howard Hughes Medical Institute's Janelia Research Campus, where I work on analyzing animal behavior using machine vision and learning. Quantitative analysis of animal behavior is a really useful tool in pretty much any field of biology that involves animals. It's particularly important in neuroscience experiments. This is because behavior is the output and the goal of the nervous system. There are two general types of experiments that we work on with our collaborators at Janelia. In this first type of experiment, we use either electrophysiology or functional imaging to record neural activity in behaving animals, and then we also record video of the animals behaving. And our goal here is to understand the relationships between this neural activity and the behavior that it produces. In the second type of experiment that we work on, we use tools such as optogenetics to be able to manipulate the neural activity. What I'm showing in the top image here is an image of a fly brain, and the neurons that are being shown in green are the ones that we're able to use optogenetics to activate. So in this bottom video, this light in the bottom left turns on when we are activating these neurons. And what we'd like to be able to do is look at how the behavior of the animals changes when we turn on these neurons to be able to understand the role of those neurons. So if you watch this video again, you can see that all of these flies start walking backwards when we turn on this light and we are activating these neurons. And that allows us to infer that these neurons that are shown in green here are involved in the control of backward locomotion. Behavior analysis is not just a tool, though. It's actually one of the fundamental questions in biology, to understand how simple animals like fruit flies are able to efficiently solve problems that are currently outside of the abilities of even the most complex AI systems. So some of the questions that we are interested in our lab is trying to understand and figure out what the structure, the vocabulary of behavior is, to understand what the rules and the heuristics that animals are following to navigate their worlds. So we use video to collect information about the behavior of animals. Video is inexpensive and easy to obtain and non-invasive. But it gives us this relatively high-dimensional information about the behavior of the animal. So this is a 1,000 by 1,000 image. That means that we have a 1,000,000-dimensional time series describing the behavior of these animals. And what our lab works on is trying to be able to to figure out a way to convert this video into a quantitative, low-dimensional, and interpretable description of the behavior of the animals. So one of the questions that we think about is, what should this representation be? And then the more standard computer vision question of, once we know what the representation is, how do we automate its computation from the video? So let's think about this question of how we should represent animal behavior. One of the dichotomies that we face is whether to use a discrete representation. For instance, we could say that there are 3 different categories of behaviors that flies do: walking, stopping, and turning. Or a continuous representation of behavior. So for instance, we could say that the properties of behavior that really matter are the speed of the animal and the distance to another animal. If we end up using a discrete representation, where do we come up with these different behavior categories? We could ask biologists to define them. Biologists have been looking at animal behavior for centuries, so they should have some good ideas of what the behavior categories should be. And if we use human-defined behavior categories, we can automate their computation using supervised machine learning. On the other hand, we could turn to unsupervised learning. And to do this, what we need to do is to mathematically define what a good representation of behavior is. So we need to be able to say that the 2 properties that really matter in defining whether 2 behaviors are similar to each other are the speed of the animal, say, and the distance to another animal. If we know quantitatively, mathematically, what it means for 2 behaviors to be similar, and we can use techniques like clustering, for instance, k-means clustering or dimensionality reduction, to learn a representation of behavior. Both of these approaches have their ups and their downs. They both require making certain types of assumptions. And what I'll do today is I'll talk about our work in both of these fields. I'll start by talking about some of our work in supervised machine learning. And here mainly we're taking pretty much off-the-shelf algorithms from computer vision and machine learning and applying them and making them usable for these applications. And then I'll talk about some of our very preliminary work trying to delve into this unsupervised machine learning and discovering structure automatically. And what I thought would be relevant for this workshop's topic is to just give an overview of some of the lessons that we've learned in terms of trying to get these types of approaches to work for our applications, and pointing out some of the problems that I think machine learning and computer vision research could really focus on to make progress. So I'm going to start with talking about behavior categorization. And our goal in behavior categorization is to be able to say at every given time point what each animal is doing. So for instance, in this video, we've come up with 15 different behavior categories, and we're annotating here what the behavior of this one fly with the triangle is at every time point. To allow biologists to train their own behavior classifiers, we've developed this program called JAABA, which stands for the Janelia Automatic Animal Behavior Annotator. And what this screencast is showing is me training a wing grooming classifier. So this top row here of this top timeline here shows the manual labels that I'm entering. So I'm labeling that the fly is grooming its wings for this subset of frames here. And then I'm going to label that the fly is not grooming its wings for another subset of frames. And you'll notice that I'm leaving a small gap of frames here where it's hard for me to say whether the fly is or is not grooming its wings. And what we've done is tried to make the user interface such that it's very easy to label a diversity of data, as well as it's easy to train new classifiers. So you can train a classifier within about 30 seconds. And look at the predictions of that classifier in this bottom timeline here. So you can look at the predictions in real time and then find frames where you disagree with the predictions of the classifier and label those. So this is a form of active learning. So we made a lot of mainly user interface optimizations with this application. And that's primarily based on a few observations that we made as we tried to solve this problem. So first, we admitted that we didn't know what all the behavior categories are, and we wanted biologists to be able to define their own. So we made usable software where the biologists could train these classifiers themselves. Next, we had to make some decisions about how to label behavior. So at the time when we started working on this project, the standard was to kind of densely label every frame in a video. And this didn't work out so well, particularly when you have these kind of long-tailed distributions where some behaviors are very rare. And so to be able to get enough labeled examples of these rare behaviors, you had to label kind of a tremendous amount of data. So instead, we allowed biologists to label whichever frames that they wanted to label, and they could easily scroll around in these videos to find positive examples of the behavior. Another thing that we noticed is that biologists are often unsure about the right label in certain frames. And instead of just making a decision, they spend all of their time agonizing over these ambiguous frames. And so what we ended up doing is just saying that the biologist should not label those frames. They should not spend the effort on giving us labels that were often very noisy. And the third observation that we made is that multiple behaviors can happen simultaneously. So that this is one of the reasons that we went with this one versus all annotation, where every classifier is trained to either be this behavior or not this behavior. The other thing that we found was really important was to make the training interactive. And this is because the data sets that are important in biology are incredibly non-homogeneous. So just to give you an example, one of the experiments that we had in mind when we were developing JAABA was this neural activation screen that we were performing, where we created 2,200 different genotypes of flies. And each of these different genotypes allowed us to manipulate the neural activity in a different sparse subset of neurons. And this produced wildly different behavior phenotypes. So for instance, activating these neurons shown in the top here produced a lot of courtship. These ones in the middle here produced a lot of jumping. And thus you could see that if we just trained on one of these types of genotypes, and then tried to apply it to a different genotype, our classifiers wouldn't work particularly well. And so it was really important for us to have this kind of interactive training where we would train on some genotypes, apply the classifier to some test data, find mistakes that it was making, and repeat. So we've been following a similar paradigm with tracking parts of animals, or pose tracking. Here, the goal is to be able to accurately estimate the positions of user-specified body parts in every frame. And again, we wanted to make it easy for biologists to track whatever body parts are important for their experiments in the constrained settings of their experiments. So we've again made one of these usable interfaces, and we've kind of thought about trying to make it easy to efficiently annotate diverse data. So here's a screencast of Alice Roby, a scientist in our lab, training a fly pose tracker. Through this interface, she can quickly annotate a frame and look at surrounding frames in the video to get the temporal context needed for accurate annotation. She can also navigate to new videos or add new videos or to different animals within the same video. One of the things that we've done is to make the training something that biologists can apply themselves. And as you all know, with deep learning techniques, there are a lot of hyperparameters that need to be tuned for each different data set. So we've tried to make the parameters something that biologists could understand by having visualizations of these, for instance, with these data augmentation parameters. We've also put the training in the hand of the biologist, so they can click this train button here and launch training either locally or remotely on a cluster or the cloud and monitor the progress. So that's what's being shown here. In this plot up here, you can see the loss decreasing as iterations of training go on. You can also use that tracker, that partially trained tracker, while it's being trained to be able to look at the performance of the classifier, of the tracker, and see if there are other new types of training data that need to be annotated. And that's what's being shown here, is applying the tracker that's only been trained here for 4,000 iterations to a small subset of video frames. So to implement this, we realize that pose tracking is an active field of research, and we've designed our system with this in mind. So the exact architecture and training algorithm that we're using is one module of the software. So for instance, we've implemented a bunch of different algorithms, including LEAP and Deep Lab Cut, which are commonly used for animal pose tracking, as well as Open Pose, which is used in human tracking, and our own algorithm, mixture density networks. This is really easily extendable and something that we hope to be able to, as new algorithms are developed, add them to APT. So basically any type of TensorFlow training code can pretty easily be integrated into our software. We've made our data grabber, which includes the data augmentation, something that is a module that you can just call from within your TensorFlow code. The other thing that we've spent time on is trying to make it so that biologists who have varying computational resources can run this code. So as you all know, you need a GPU to be able to efficiently train these deep learning models. And so we allow different operating systems, so Docker for Linux, Conda for Windows, as well as cloud computing with AWS or on a cluster. And so all of that is implemented within our system as well. So people have been working on pose tracking for quite a while, and it's working pretty well enough to actually do some biology with the outputs of these systems. However, it's still not at the level of human performance. So this gray line here is our intra-annotator error, and you can see that even as we are adding a large amount of training data, our performance is not approaching human. And I think this is because all of our annotation, all of our automatic annotation, I should say, is being done on individual frames. So things like Open Pose, for instance, just input a single frame and try and do the prediction there. And from a single frame, it's quite hard to know exactly whose leg belongs to whom or which leg is which. And that video did not play. But pretend this video played. Let's see. Nope, that video is not going to play. So if you could see this video playing, what it would show you is that temporal context is really important. It makes it much easier to distinguish which leg is which. And it's really important for the annotator in being able to solve this problem. So we hope that computer vision researchers, and we ourselves are working on this as well, will concentrate on really using these large spatiotemporal volumes. And this requires both new algorithms for doing this, as well as data sets where it's actually vital to accuracy to be able to use these large blocks of temporal information. Okay, so in my last few minutes here, I'm going to switch gears and talk about a very recent project that we've been working on, where we're trying to use unsupervised machine learning to discover new behavior structure. So here we've been looking at the problem of behavior forecasting. So in behavior forecasting, our goal is to be able to predict the future behavior of an animal given its current environment and past behavior. So this is an important problem in self-driving cars, for instance, because one wants to know whether a pedestrian will likely intersect a car's planned path. But it's actually, I would say, an even more fundamental question in biology, because if we have a model, a machine learning system that can predict what an animal is going to do, then we have a really good model of this complex biological system as a whole. And so if our model truly behaves like the animal, then understanding the animal reduces to understanding this machine learning model. And so we can learn a lot from using this kind of machine learning. So we have a couple of questions that we have to answer for this to actually work. Does the machine learning model actually behave like a real animal? And then this other question, which is becoming a big field within machine learning of explainable machine learning. So how do we interpret this machine learning model, which itself is a very complicated thing. So to this end, we've been training recurrent neural networks that input a representation of the animal's current environment and learn how to represent all of the past information that's relevant to predicting the fly's future behavior. So what we've been spending most of our time on so far, because we're just getting really started on this project, is trying to figure out whether our machine learning models really are working like a real fly. So one thing that we can do, which is kind of the standard thing to do, is to look at the log likelihood of our test data given our model. But this is just a unitless number. If we get something like 57,000, is that good? Is that bad? It also requires that it assumes that the assumptions of our model are correct. And finally, this model is only trained to predict one frame into the future, whereas we want to see trajectories that are realistic looking. So we can, to use this single frame prediction, we can turn it into a multi-frame prediction by using the single frame prediction to predict one frame into the future, feeding in the resulting environment that corresponds to that, and re-predicting. This allows us to create simulations of a single fly, or we could actually do this for all of the flies in the environment and get simulations of multi-agent behavior. So the first thing that we do as engineers is the eyeball test. We can look at whether we can fool biologists with these simulations. So one of these videos is from real flies, and the other three are from different machine learning models. And to me, I would say that except for the linear regression model, these two models here, the recurrent neural network and a convolutional network, often will fool me. They often look close enough to real flies. This, of course, requires a biologist to be in the inner loop. So another approach is to train a discriminator network that can tell real from fake trajectories. And then the final approach that we've been exploring is to look at population statistics collapsed over time. For instance, histograms of the speed of the animal or the distance to another animal to find deficiencies or biases in our models. So, of course, the problem I haven't talked about yet, which we're just, again, getting started on, is once we've convinced ourselves that our models behave enough like real animals to be of interest, how do we learn new properties of the algorithms that the flies use to navigate their world and how they're implemented? And then how and why might that be the case? So to solve this, we're combining techniques from explainable AI and experimental biology, where our model is the experimental subject. So I'm going to end there and thank my lab and our collaborators, and thank you for your attention.